I have a few more points to go through before we completely wrap up, and that begins with thanks to all of you for the participation that helped us get to the point where we are today to make some progress towards addressing the big challenges that we face in cancer care in this country. Just going back over briefly some of the main points from today and some of the next steps that are going to come out of this meeting. First, we started off by identifying some opportunities to significantly reduce the cost of data collection and increase participation in clinical trials, a big challenge right now, by reducing the burden of participation on both patients and clinicians. One way to do that is to reduce the collection burden for data that does not have much value, low value or no value, and by working together, our first panel with a lot of leadership from ASCO found some specific suggestions for how to do that on supplemental drug applications for data related to adverse events. This is a great example, I think, of getting to some real progress in the development science and the regulatory science that go along with it. We need to row those oars together to make progress on learning more and learning it more quickly and more efficiently. Our second panel took up some issues that had begun to be discussed last year at our last year's conference on progression-free survival, and came to the clear conclusion that there is a significant issue of discrepancy in reading progression-free survival, but that discrepancy doesn't necessarily mean bias. Rather, a differential rate of discrepancy may indicate a bias problem and a greater need for independent review. The panel went through some ideas for how to characterize that and the possibility of partial independent blind review. Rick Pazder highlighted a different implication of some of the challenges with progression-free survival discrepancy and the measure itself. Maybe it's an indication that we need to consider further issues related to the clinical significance of progression-free survival, at least in some cancers. So both the potential for bias and the clinical significance likely vary by cancer, and as our pilot work on the partial independent blind review process proceeds, another next step may be to expand this process to put progression-free survival in the context of other markers of clinical benefit. And after lunch, we came back to a very spirited discussion around the challenge of getting targeted therapies that really truly work to cancer patients who badly need them. The discussion didn't stick with generalities, although this is a topic that we hear about a lot these days. Instead, it triggered a stimulating focus on identifying key issues for specific practical analysis. I'm not going to try to summarize all that discussion here, but I can assure you that we're going to be doing some further work related to this discussion. For example, there was a lot of talk about clarifying the importance of distinguishing markers for predicting response from markers for indicating clinical benefit and focusing in on each of these important challenges. With respect to the former, for predicting response, I think it will be possible to do some more specific thinking and analysis on what to do if there is a promising marker for a targeted therapy, for predicting response to a targeted therapy. There were some arguments made that with a good marker, a phase 3 trial is a straightforward and small and not costly approach to demonstrating effectiveness convincingly, because the clinical response would be so clear if it really is a good marker. On the other hand, there were arguments that it's not so easy, and so targeted approval without a randomized phase 3 trial is necessary to provide the reimbursement incentives to bring these treatments to narrow populations of patients who can benefit. But that, of course, raises the question of how do you know it's really a marker for better outcomes from treatment rather than just better underlying prognosis, or more practically, what exactly will happen after that targeted approval occurs with reimbursement to settle this issue. And I think we can do some more work, some more analysis to clarify those two alternatives. Finally, with this most recent panel, which is a great discussion on the issue of targeted therapies, as John Niederhuber emphasized, combination therapies affecting a broader range of cancer care today, and it's going to get more complex as we have a better understanding of the entire environment of a cancer and in which it occurs in individual patients. So a framework for evaluating combination therapies more efficiently could be very helpful. The FDA has a foundation to build on here, and as Janet pointed out, this is not, what we're not talking about here is not fixed combinations like OTC drugs or some of the cardiac combination drugs, but rather combination profiles of treatments, combinations of therapies. And these combination regimens that are expected to have significant clinical benefits that outweigh their potential for increased risk is an area where FDA is planning to develop guidance. It may not be just limited to cancer, but that's a point about how important further work in this area will be. So building on today's paper could be very helpful in moving this process forward. In doing so, we need to address the issue of elevated toxicity risk. And as some of the suggestions just a few moments ago from Bob and others highlighted, it may be possible to do this with some short-term endpoints on looking at therapy A and therapy B separately versus A plus B to do the dose finding and the dose toxicity, the dose response finding, perhaps with short-term endpoints. I'm not sure if that meant, Bob, whether a less than fully validated marker might be worth considering at that stage, but that's something we definitely would like to pursue.